Dear students, in this module we're going to talk about the visualization technique that can be employed to visualize protein structures. You know that protein structures are formed as a result of a folding process in which a polymerized chain of amino acids takes a 3D form. As a result of this folding, the phi and psi angles are formed along the backbone of the protein. You can measure these psi and phi angles and then plot them in order for you to construct the backbone structure of the protein. However, if you want to plot each atom within the backbone, you need to plot the atomic positions of the atoms in the backbone. So, towards looking at the structure of a protein and the atoms inside the backbone of this protein, you need to have specific positions for each atom along the backbone of the protein. To simplify the situation, you can simply take one atom along the backbone and you can plot it repeatedly, and that can be a representative of the backbone structure of the protein. During the protein structure plotting and visualization, what we do is we take up the alpha carbon from the backbone of the protein and we take the coordinates, that is x, y and z positions of each alpha carbon within the entire backbone, and we plot them. So that is how we visualize the proteins. You know that the backbone consists of carbons and nitrogen. Let me draw this backbone for you. So this is an amino acid for you, and this is the location of the R group. So this carbon here is called the alpha carbon. So you know that there are multiple alpha carbons within the backbone because there are multiple amino acids that are lining up in the backbone of a protein. So given several alpha carbons followed by another carbon, we typically call it the beta carbon, followed by nitrogen, alpha carbon, beta carbon, and so on. So essentially, you can simply collect the coordinates of each alpha carbon along the backbone of the protein, and you can plot these positions to form the backbone of the protein. So as I just mentioned, to visualize the protein, the first step is to construct the backbone in 3D. To construct the backbone of the protein, you simply take the positions that are occupied by alpha carbons, or simply Ca, carbon alphas, and then using PDB, you can retrieve the x, y and z coordinate for each alpha carbon and construct the backbone. I will show you an example PDB here, and as you know, the C alpha attribute here, here and here, it shows you the coordinates for the alpha carbons along with their position. So you have number 2 alpha carbon in this protein that is taking up these coordinates. You have another alpha carbon here, you have another alpha carbon here, and therefore you have all the coordinates that are available for each alpha carbon, and therefore you can construct the backbone now. So in conclusion, the protein structures can be visualized by simply taking the coordinates of the alpha carbons that are there in the PDB file of a protein. So once you take the coordinates of all the alpha carbons, these will give you the x, y and z positions of each alpha carbon. You simply take these values and you connect them, and you have the backbone of the protein. Now you need a tool to visualize this backbone.